Researchers have identified two types of cells in our brains that are involved in organizing discrete memories based on when they occurred. This finding improves our understanding of how the human brain forms memories and could have implications in memory disorders such as Alzheimer's disease. The study was supported by the National Institutes of Health's brain research through Advancing Innovative Neurotechnologies BRAIN Initiative and published in Nature Neuroscience. This work is transformative in how the researchers study the way the human brain thinks, said Jim Gnadt, Ph.D., Program Director at the National Institute of Neurological Disorders and Stroke and the NIH BRAIN Initiative. It brings to human neuroscience an approach used previously in non-human primates and rodents by recording directly from neurons that are generating thoughts. This study, led by Welly Rudeshauser, Ph.D., Professor of Neurosurgery, Neurology and Biomedical Sciences at Cedars-Sinai Medical Center. We live our awake lives as one continuous experience, but it is believed based on human behavior studies, that we store these life events as individual, distinct moments. What marks the beginning and end of a memory? This theory is referred to as event segmentation, and we know relatively little about how the process works in the human brain. To study this, Rudeshauser and his colleagues worked with 20 patients who were undergoing intracranial recording of brain activity to guide surgery for treatment of their drug-resistant epilepsy. They looked at how the patient's brain activity was affected when shown film clips containing different types of cognitive boundaries, transitions thought to trigger changes in how a memory is stored and that mark the beginning and end of memory files in the brain. The first type, referred to as a soft boundary, is a video containing a scene that then cuts to another scene that continues the same story. For example, a baseball game showing a pitch is thrown in. When the batter hits the ball, the camera cuts to a shot of the fielder making a play. In contrast, a hard boundary is a cut to a completely different story. Imagine if the batted ball were immediately followed by a cut to a commercial. Jia Zheng, Ph.D., postdoctoral fellow at Children's Hospital Boston and first author of the study, explained the key difference between the two boundaries. Is this a new scene within the same story, or are we watching a completely different story? How much the narrative changes from one clip to the next determines the type of cognitive boundary, said Zheng. One group, called boundary cells, became more active in response to either a soft or hard boundary. A second group, referred to as event cells, responded only to hard boundaries. This led to the theory that the creation of a new memory occurs when there is a peak in the activity of both boundary and event cells, which is something that only occurs following a hard boundary. One analogy to how memories might be stored and accessed in the brain is how photos are stored on your phone or computer. Often, photos are automatically grouped into events based on when and where they were taken and then later displayed to you as a key photo from that event. When you tap or click on that photo, you can drill down into that specific event. A boundary response can be thought of like creating a new photo event, said Dr. Rudeshauser. As you build the memory, it's like new photos are being added to that event. When a hard boundary occurs, that event is closed and a new one begins. In the first, the participants were shown a series of still images and were asked whether they were from a scene in the film clips they just watched. Study participants were more likely to remember images that occurred soon after a hard or soft boundary, which is when a new photo or event would have been created. It turned out that they had a much harder time choosing the correct image if the two occurred on different sides of a hard boundary, possibly because they had been placed in different events. These findings provide a look into how the human brain creates, stores, and accesses memories. Because event segmentation is a process that can be affected in people living with memory disorders, these insights could be applied to the development of new therapies. In the future, Dr. Rudischauser and his team plan to look at 2 possible avenues to develop therapies related to these findings. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.